based on metabolic dysfunction and damage to hepatocytes. And specifically for HCA Asia, this is a recent study from 2019. It appears NASH is the fastest growing cause of HCC. The incidence is actually very similar between men and women, so very different from Hep B related HCC, where men get it about 4 times higher proportionality compared to women. And you can see in terms of the rapid increase, specifically in India as well, this is just in the last 9 years. So the proportion of HCCs that are related to NAFLD is on a rapid rise. So therefore, again, becomes really important for us and pertinent for us to study this. Somewhat controversial, NASH HCC is also appeared to be less responsive to immune checkpoint blockade, although the jury's still out there. But fundamentally, the hypothesis that we are working on, and this is again collectively with the liver seed network as well, is that we think that liver damage, so whether it's metabolic dysfunction associated or viral hepatitis, for example, may activate regenerative pathways that can reprogram the liver tissue microenvironment in somewhat of a pro-tumorigenic niche. But obviously there are multiple questions that we want to ask. What are the mechanisms that drive these distinct etiologies of liver disease progression? So whether it's viral or NAFLD-NASH to HCC, how do these distinct etiologies relate to altered cellular organization or neighborhoods and their ecosystem? How do they modulate therapeutic response? And better understanding of this, can we then target these mechanisms as early preventative intervention strategies, which has now become the major thrust of my lab. We want to be able to reverse NASH, NAFLD, reverse chronic Hep B, for example, so that we don't get to or minimize the risk to progression to HCC. Once you get HCC, it's already kind of late. And this is a slide that Sham shared earlier yesterday, and this is what we are planning to do. And I'm showing you this again mainly so that it's also an invitation for anybody who's interested in looking at these early trajectories and etiologies, the distinct etiologies of liver disease progression to either NASH or non-NASH HCC. Please come and have a chat with me. But what we are doing currently is basically generating a multimodal, so single-cell RNA-seq, ATAC-seq, as well as flow cytometry-based assessment to generate a spatiotemporal atlas of both liver disease progression as well as regression. We already have some really beautifully curated samples, patient cohorts with distinct stages of NAFLD and early NASH and late NASH, and then their progression to HCC. We also have certain cohorts where we have pre-post samples from patients who were under diet and physical activity regimen, so where they show reversal of NASH, for example. So lifestyle and dietary interventions. So the idea is to generate a comprehensive atlas, not just with single-cell multiomics, but also getting into some of the DESI MSI, which is a metabolic spatial imaging technology. And then again, in parallel, we're also looking at some of the mouse models of liver disease progression for functional studies. However, for the rest of today's talk, so that's all data being generated, and hopefully I can talk to you about it in a lot more detail the next time we meet. But I'll talk to you a little bit about some of our more recent work. This is currently a preprint and under revision of single-cell analysis of chronic Hep B functional cure. Functional cure as it is defined by the loss of S antigen. So why are we interested in Hep B? Well, viral hepatitis and HB can activate just about all 10 hallmarks of cancer, and in fact, one of the predominant cause of HCC, so 40%. Even though NAFLD-NASH is very rapidly on the rise, this is work led by 2 extremely talented postdocs, Narmada and Ati, together with Niranjan, who's a bioinformatician, and Lim Sengi, who's our clinical collaborators. So why do we care about understanding the mechanisms of functional cure, that is loss of S antigen? And as you can see in this graph, and these are again based on EASL guidelines, there are 4 phases of chronic Hep B, and then functional cure as defined by the loss of S antigen. But the reason we want to understand is that, well, firstly, it's a clinical endpoint that the clinicians want to reach in terms of achieving functional cure, and the reason is that it minimizes your progression to HCC. However, the mechanisms that are driving functional cure in patients who are treated with nucleoside analogs combined with interferon alpha to elicit an immune response are really poorly understood. So we really want to understand those mechanisms that underlie long-term functional cure as a means to then minimize the risk of progression to HCC. And so that's what we did. So this is just the workflow that we have. So it's about 22 patients, 15 with chronic Hep B, 7 that achieve functional cure after treatment with antivirals combined with interferon. We generated single cell data from core biopsies as well as matched PBMCs, and also analyzed by flow-based immune phenotyping and then followed by spatial validation. So this is now in Bar Archive, you can read more about it, but hopefully you'll see it soon. So I'll just focus on some of the key observations that we've made in terms of mechanisms or immunopathological cell states associated with functional cure, and what we could learn about how can we affect that functional cure in patients. So first, in functional cure patients, so the patients that have lost S antigens upon treatment, they exhibit a remarkable switch from an immune tolerant or immune suppressive to an immune active environment. So you lose these clusters of Tregs, which are regulatory T cells, as well as lose these clusters of exhausted CD8 positive cells. The other really interesting and very surprising thing is that we also see this gain of this red cluster, and these turn out to be CD4 CTLs. And this is surprising because these are CD4 T cells, helper T cells that acquire properties of innate immune-like cells, including gain some of the markers such as NCR3, but they also gain cytotoxic or cytolytic abilities in terms of expressing granzymes, perforin, and TNF. We could validate this with functional, with flow cytometry. Again, you see these CD69, which are the tissue-resident populations of CD8s, now stratified with different levels of S antigen. So red is high levels of S antigen, orange is moderate, green is low, and blue is functional cure. Let me see if this pointer works. Not really. Can you make this pointer work? All right, thank you. Yeah, hopefully this is better. So you see this loss of the exhausted populations, and moreover, you also see gain in these CD4 T cells that are gaining this marker KLRG, which specifies these terminally differentiated antigen experienced CD4 CTLs. They also express these markers, and we validate that with flow cytometry, as well as CyTOF. And this is again looking at patients with very high levels of S and low levels of S. You can see about a 2.5 to 3-fold increase in granzyme B and perforin in the CD4 KLRG population. So that was the first surprise, right? The second thing that is really interesting, and this is functionality of these CD4 cells. So we basically stimulated these T cells with pooled peptides representing different viral proteins, including X, core, Pol, and S antigen. And what I really want to focus your attention to is that the HBS-specific CD4 T cells, they appear to be present at much higher frequencies than the CD8 S-specific CD8 T cells, right? Suggesting that the dominant S peptide-specific response is actually coming from a CD4 effector, not CD8s in functional cure, suggesting that these CD4 CDLs may be actually playing a really important role in not just driving functional cure, but even maintenance of that functionally cured state. And you also see increased multifunctionality in terms of interferon and TNF alpha expression in these S-specific CD4 T cells. And this kind of corroborates somewhat some of the recently published data from Georg Lauer's group. They used tetramers basically against MHC class 2, where they showed that these Hep B-specific CD4 T cells are reliably found in the S-negative patients and almost rarely found in the S-positive. Right. So one thing that we started thinking about, well, then what are these CD4 CTLs recognizing? Because these would be interacting with MHC class 2. And the other major surprise that we find in the functionally cured state is this emergence of these MHC class 2 expressing hepatocytes, suggesting these hepatocytes maybe gaining accessory APC, antigen presenting function, upon interferon alpha treatment. Now, a couple decades ago, actually, this has been shown in vitro that interferon can actually induce an MHC class II switch. What we think is that the patients who have undergone this nucleoside analog treatment combined with interferon, they're causing this switch in MHC class II in hepatocytes, which are then recognized by the CD4 CTLs to take them out. However, there lies a conundrum because we are talking about functional cure, and at least clinically speaking, you were saying, well, I mean, the S loss, there is S loss, right? So at least in the serum. So what exactly are the hepatocytes actually presenting, even if they were to present viral peptides, for example, because this is an S loss case. But what we went back and started looking at specifically in a more granular fashion, the different viral parameters in these functionally cured patients. And what you can see with digital droplet PCRs, as well as single-cell RNA-seq actually. So we took the unmapped reads and mapped it to the Asia-specific genotypes of the virus, and we see a 50 to 100-fold reduction in the production of virus, but the virus is definitely still being produced in the functionally cured state. So you have levels of cccDNA, Hep B DNA, the HBV 3.5 kb mRNA as well. And then if you look at the normalized ratio of the mRNA to the amount of cccDNA, which is transcribing this viral DNA, you see the transcription rate is more or less same, very similar between chronic Hep B and functional cure. But we could now see the same thing with the single cell profiling as well. So this is basically mapping the unmapped reads otherwise down to the viral genotype, suggesting that even though there is very low levels of the virus in the functional cured state, they could still be now presenting the native peptides, as also suggested by Georg Lauer's group, and eliciting a CD4 CTL response. So a couple key take-homes: that the loss of S antigen detection in serum is not necessarily cannot be equated to loss of S transcript production in the hepatocytes, and the reduced viral load upon nucleoside therapy combined with the interferon-mediated activation of MHC class II and the IL-2 driven differentiation of CD4 CTLs. There's a lot more details there that I'm skipping over because of time constraints, but eventually would result in a better outcome to chronic Hep B. So just a working model for the CHB resolution is that we think that in the state of chronic hepatitis B, you have a defective priming of the adaptive immune system by a lot of pseudoantigens that are floating around, that are produced by the hepatocytes, which then results in chronic antigen stimulation and immune cell exhaustion to create this tolerant microenvironment. And upon treatment with nucleoside analogs and followed with to lower the viral load, and then followed up with interferon therapy, you can reactivate these pro-differentiation of CD4 CTLs together with MHC class II, where hepatocytes are now presenting native peptides to CD4 CTLs. And again, a really important role that I didn't really talk about here of the innate immune arm as well, with NK cells and neutrophils, we think are really important in affecting resolution of Hep B. But overall, the other point just wanted to make is that probably need to consider CD4 T cells when designing the next generation of immunotherapies. So just a couple last take-home messages or take-home slides that I will leave you with. So at this point, what we are now trying to do is kind of develop a blueprint for across time span and disease, both in health as well as disease states in the human liver. So this is basically a Skivvy-based integration of the different data sets that we have now downloaded or generated, and now we are combining them to generate these integrated maps, and we can annotate these maps. But fundamentally, we want to kind of try to see, these are the disease-associated cell states, for example, and the fibroblasts, which are not there. Perhaps, okay, this is Mazhanifa's fetal data sets, early versus late. We see very interesting changes in endothelial cells in early versus late in the fetal liver, and some of them could be represented again in situations of chronic liver damage, whether it's cirrhosis, Hep B, or HCC. Similarly, we are also developing a blueprint for healthy and disease states in using the mouse models. And again, hopefully you could see this with control diet. For example, this olive green cluster is basically a fibroblast cluster, and you see remarkable changes what's happening in the fibroblasts, as well as the endothelial cells and the hepatocytes, which are very distinct from what we see in the animals on normal chow or normal diet, suggesting that there is a remarkable remodeling of these different parenchymal and the non-parenchymal cell states, and it's really the crosstalk between these that then generate, what we think would be a pro-tumorigenic microenvironment. So with that, I'll end here and to the most important slides really of acknowledgement. Patients who were so generous with donating all their samples for research. Really the true heroes are the people who do the work. I mean, these are the DG Lab heroes, the current and the past members. I'm basically the cheerleader on the sidelines, and amazing collaborators and clinicians out there. And while we're doing all this work, we also have a lot of fun. So happy to take questions. I'll end here. Thank you.